I didn't record that part. Oh, my speaker is not working. Sorry, my speaker is not working. It was working yesterday. You're seeing that still. Okay, are you working? We'll see what happens. Okay, couple things and reminders which I forgot. I forgot to tell you last Friday. And it's probably wouldn't matter if you necessarily remember, but today is the beginning of the Barn Symposium, which is a somewhat regular event talking about the intersection of faith and science. And this year it's coinciding with the seminary. They're putting it on, and it's someone who's going to talk about creation care and justice. So I'm willing to give you extra credit because I am interested in creation care. And so if you go to chapel today, which is yes, right after this class, not a lot of notice. There's an assignment on Moodle. It's not a hard assignment, it's just some questions to answer based on what she talks about. I don't exactly know what she's talking about. So I tried to make questions that might make sense based on what I think she's going to talk about. She's talking about the story of Zacchaeus and something about trees. So after that, there's a lunch which you can go to in Ellen Stowe. It's supposed to be a student lunch, so it's really great if students show up and not just faculty members. So that's there. Chapter 10 homework is posted on Moodle. It's due Friday at the beginning of class. There's a quiz on Friday, which I did not put on there too. That will be open at the normal hours. And then our test on chapters 9 and 10 will be next Wednesday, so a week from today. Again, chemistry is that Friday, and this test will be Wednesday. Questions about any of that? Okay, so we finished chapter 9 basically before we left on Friday. So today we're starting chapter 10, which is photosynthesis. So if you thought cellular respiration was fun and detailed, wait until you get to photosynthesis. It's even more fun and more detailed. So buckle up because there's a lot. You do have that yellow sheet. We will be using it some. I confess I made the yellow sheet thinking more about cellular respiration. So the backside, we're gonna have to make some changes to because I didn't do well. But we will use that as we go along. Just some reminders. This is what we're looking at with the overall process of chapter 9 and chapter 10. So we've got two systems of equations, two systems of energy conversions. We've got the top part, which is chapter 10. This is the photosynthesis part that we'll be talking about, and the bottom part, which is chapter 9, which we talked about. Remember, energy is flowing through the system. We're starting with light energy. It's flowing through the system, used in the chloroplasts to make organic molecules, and then that's converted to chemical energy in the form of ATP, which is used for cellular work, and what isn't used effectively is dissipated as heat energy. So we've talked about cellular respiration happening in the mitochondria. What kind of organisms do cellular respiration? Somebody said something in here. It happens in the mitochondria, yes. What kind of organisms do cellular respiration? Trees. So plants and animals and bacteria and fungi and protists. Okay, we're going to talk about photosynthesis, which means we're going to be narrowing in more on plants, but I want to remind you that plants do cellular respiration. Plants have mitochondria. Plants need energy, not in the same levels that an animal does, but they still need ATP, so they're still doing cellular respiration. And again, we talked about the different forms. It can be anaerobic or aerobic, but some way to make ATP to get energy. Okay, so photosynthesis is where we are starting. We've talked about this before, but the meaning of the word photosynthesis. Photo means light, and synthesis. Syn means together, so we're putting things together. It's the process of putting things together with light. And we're going to go into extreme detail about this process and how this happens. We've talked about, again, this is happening in what kinds of organisms? Plants not fungi, not animals. Some protists, I think in lab you looked at Euglena, right? Euglena are photosynthetic. They're also heterotrophic, so they're mixotrophs. But they can do photosynthesis. Algae are considered protists. They can do photosynthesis. Actually, yeah. And some prokaryotic cells can do it as well. I'm putting a star next to them. Why would I think that prokaryotic cells are a little different than the other two? What do they not have? They don't have mitochondria and they don't have chloroplasts because they have no membrane-bound organelles. So everything that we talk about with them, with either cellular respiration or photosynthesis, is happening in the plasma membrane. So their plasma membrane serves the function of the mitochondrial membranes or the thylakoid membrane. So photosynthesis happens in plants, protists, and some prokaryotic cells. Organisms that do photosynthesis are what we call autotrophs. Auto meaning what? Self. If your car's on autopilot, it drives itself. I guess we just call it self-driving now, but autotrophs are self-feeders. So they're using a process to be able to feed themselves. When we talk about photosynthesis, we're specifically talking about photoautotrophs. So they're using light as the energy to drive this process. To drive the process of making organic molecules, they use light. So that's what we're focusing on this chapter because it's photosynthesis. There are some, and this is the majority of them, there are some that are called chemoautotrophs. That instead of using energy from light, they use energy from hydrogen sulfide. These are really weird little prokaryotic cells that live in the hydrothermal vents of the ocean. So really deep, high temperature, high pressure, and no light, but they're able to still sustain themselves. This is the basis for everything. This is the basis for all food chains. So these guys, whether they're photoautotrophs or chemoautotrophs, are what we call producers. They are the producers because they are making organic molecules out of inorganic molecules. So they're taking raw materials from the environment and they're able to turn them into high energy organic molecules. And again, they're producers. This requires some input of energy, and we're talking about photoautotrophs, so that input of energy comes from light. On the other side, you see these, I forget what the name of this is. I just had it. It's two-spotted. There's some weird name for them. We see these beetles eating leaves. They are not autotrophs. They are what? Heterotrophs. They are not self-feeders. They are other feeders. Meaning they have to eat something else to get their energy. Okay, who falls into this category? Animals. What else? You used one in lab last week, Tuesday, Wednesday, people. You'll use one in lab this week, Thursday, people. What did you cut up? Mushrooms. So fungi are also heterotrophs. Obviously, mushrooms aren't out there like hunting down squirrels and eating them, although that would be entertaining to watch. These are a specific kind of heterotrophs called decomposers. So they release enzymes into the environment to break things down so they can bring those raw materials in. So they're still feeding on other stuff. It's just not like chomping down and eating other stuff. So fungi are this, a lot of prokaryotic cells are this. A lot of bacteria function as decomposers, and some protists, because protists just do everything. And again, they're not producers, so whether you're heterotrophic eating something or you're a decomposer, you're considered a consumer. All life on Earth, pretty much, because in biology, we are really skeptical of the word all. All life on Earth, pretty much, is dependent on this process of photosynthesis, is dependent on photoautotrophs. So we are completely dependent on photoautotrophs to provide the organic molecules that we need. Even if you don't ever eat a plant a day in your life, you're a carnivore only, you're still dependent on plants because something that you eat eventually gets to the point where it ate plants. So all life, except for not all, because the chemoautotrophs. We're dependent on photoautotrophs, not just for our food, not just for the organic molecules we need, but also for what else? What else is produced in photosynthesis that we need? Oxygen. So we are directly or indirectly dependent upon photoautotrophs for the food and oxygen we need. This is a really important process. Okay, and again, we're going to start with the big picture. So on your yellow sheet at the top, you can write in this, which is the equation for photosynthesis. This equation that you see here. This is our basic equation for photosynthesis. So I'm again trying to remind you where we are in the book. Chapter 10, section 2 is talking about how we convert light energy into chemical energy. Now, photosynthesis is the process of taking this light energy and making it into chemical energy. So keeping the big picture in mind, then we're going to break it down into all the little detailed steps. We know that the equation for photosynthesis says we bring in energy from where? So light energy. When we talk about energy in the cellular respiration equation, we're talking about ATP. And we're talking about energy in the photosynthesis equation, we're talking about the sun. So light energy. And what are our raw materials? What are our reactants? CO2, which is what? Carbon dioxide, is it organic or inorganic? It's actually technically inorganic. You're right, it has carbon in it. It's this one little exception to the rule, and it doesn't have any hydrogen in it. So really, an organic compound has a carbon hydrogen bond. So it's technically inorganic. Where does it come from? What's the source of carbon dioxide for plants? Where do they get it? All around them in the air, yeah. So the source of this is the atmosphere. And it's going to bring it in through the stomata, which we'll talk about in a minute. The other reactant is water. Where is that coming from? Where do plants get it? From the ground. They suck it up through their roots generally. Which really came from the atmosphere there too, but that's okay. Okay, those are the raw materials. You can take air and water and stir them together all you want while shining a light on it, and you're not going to get sugar out of them. Okay, plants have an elaborate mechanism, the structure of which allows them to function to make the products, which are glucose. And really, we should be writing this more generically because it will tell you as we get into this a little bit more, it's really a lie when we say it makes glucose because it doesn't directly make glucose. Not a lie. It's a temporary deception to make it easier to understand. Okay, so it's making some kind of carbohydrate and oxygen. So again, this is a redox reaction. Redox meaning what? What are we gaining and losing? Electrons, and we know that oxidation is loss and reduction is gain. Leo the lion says grr, or oil rig, whichever one makes you happy. So in this case, we have carbon dioxide over here. Carbon dioxide is going to be reduced, which means it's going to gain electrons. As it gains electrons, it becomes this carbohydrate. You'll notice it's also gaining something else. What? Here it's CO2, and over here it's C6H12O6. So what else does it have to gain? Somebody said it. And hydrogen ions. Remember, in cellular respiration, every time we move those electrons down the electron chain, we could move a hydrogen ion. Same kind of idea here. We're going to move electrons and move hydrogen ions at the same time. They move together. So it's going to gain electrons. It's going to be reduced by gaining electrons and hydrogen ions into a carbohydrate. Water, on the other hand, is going to become oxidized, which means it's going to do what? It's going to lose electrons. And you'll notice it's turning to oxygen. So what else is it losing? Hydrogen. So it's losing those hydrogen ions also. Up at the top here, you can see this is showing you the reactants and where each of these molecules is going to go, where each of these atoms is going to go. So the carbon from carbon dioxide is going to go to glucose. The oxygen is going to go to both the glucose and water molecules. This water that is broken down is going to donate hydrogen to the glucose and hydrogen to the water and oxygen to the oxygen. So the oxygen that is produced here is ultimately coming from water. The water that's produced here is coming from both water and carbon dioxide. And the C and the H and the O are coming from both carbon dioxide and the water. You'll notice we didn't even write water in there. There is actually water on this side. Also, we're putting in 12 oxygens and 12 waters and getting out 6 waters, but we're not really worrying about that. But that's why it's showing water as a product as well. So that's our overview. Again, this is a reaction that is converting light energy to chemical energy in the form of bonds in organic molecules in the carbohydrates. Does this equation look familiar to you overall? If you're just looking at the overall equation, what does it look like? The opposite of what? It's the opposite of cellular respiration. And that's why we kind of fudge it a little bit and say it's glucose, because it's easier to see that it's related to. And we're going to see a lot of opposites and a lot of similarities. Okay, a lot of similarities in the way things work, but working in the opposite direction, but also some differences. So as we're going along and talking about these different steps, think about, how is this similar to cellular respiration? How is this different from cellular respiration? Right, that's something that you need to be thinking about, and we'll also discuss as we go through. We're not even ready to start though, because we're going to talk about structure of chloroplasts again. Just like we talked about the structure of the mitochondria when we started cellular respiration, we're going to talk about the structure of the chloroplasts. We have not really talked about plants, and people tend to not be necessarily as familiar with plants unless you're really a plant lover. We don't honestly even touch plants that much in Gen Bio 1 or Gen Bio 2. Gen Bio 3 is where we really get into plant structure and function. In lab last week, we looked at, in Gen Bio 3, we looked at the structure, sorry, of a leaf. We looked at the cross section. So you will see that if you're a biology major and you get to Gen Bio 3. If you're a biochemistry major or biomedical sciences major, exercise science major, you won't take Bio 3. So this will be your plants right here, all in one slide. Okay, so in a plant, most cells have chloroplasts, but chloroplasts are mostly functional in leaves. So anything that's green in a plant has chloroplasts, but chloroplast function of carrying out photosynthesis is happens mostly in the leaves. This is the time of year where we become very aware of this function. I don't think we're at peak yet. I don't know if any of you live where you're at peak for leaf changing. We're not quite there yet, but we're starting to see the color changing, and we'll talk about why that happens. And next week in lab, you'll look at some different colors of leaves and what they look like in terms of what pigments are still there. Again, the structure of the chloroplast and the structure of the leaf helps this to be functional. So this is the cross section of a leaf. So this is the, is this not right? There we go. This is the upper epidermis and this is the lower epidermis. Inside, we've got all these cells in this layer called the mesophyll, and this is where most of the chloroplasts are. So they're located in the mesophyll layer. There's two different kinds of, you'll notice the top ones are more straight up and down, and these are kind of a mess. So this is called the palisade mesophyll, and this is the spongy mesophyll. Little bonus information for your life. This is where the chloroplasts are. Notice underneath, so this is the lower epidermis. You see these look like coffee beans to me. Maybe I need more caffeine in my life. Maybe I don't need more caffeine in my life. These look like coffee beans. They're called the stomata. Stomata just means little mouth. Okay, you eat with your mouth, but what else do you often do with your mouth? You can also breathe with your mouth. And this is how the plant is going to move carbon dioxide in. I'm probably going to need a different color. So carbon dioxide can enter through these stomata, these little openings, these little mouths, and oxygen will leave. So this is where gas exchange occurs in a plant, through the stomata. And they can open and close depending on different weather conditions, especially like when it's hot or windy, they'll close so that the plant doesn't also lose water, because sometimes water exits through the stomata. So this is where the exchange of gases happens. This is where the carbon dioxide goes in for photosynthesis and the oxygen from photosynthesis comes out. You also can see the vein here. The vein in the leaf this is where water is coming from the roots. It moves through the veins using what force will move it? What forces will move it up the plant through the veins? Way back to chapter 2. Cohesion and adhesion. The water molecules stick to each other and they stick to the sides of the veins, and that'll bring it up the plant. And then it can diffuse in and out. So carbon dioxide and oxygen and water all can move through diffusion from high concentration to low concentration into the cell. So in a mesophyll cell, there are lots of chloroplasts. I have a number. There's about that's a little more impressive to me than 30 to 40%. So there's a lot of chloroplasts. Within these cells, my internet connection is unstable. You can see some things that might look familiar when you think about mitochondria structure. Okay, what do you notice about this that might remind you of a mitochondria? How many membranes do you see? 2. How many membranes does a mitochondria have? 2. So we've got an outer membrane and an inner membrane, just like you would see in a mitochondria, but we've got an extra bonus in here too. Within this inner membrane, we can see all these little stacks. Some people say they look like stacks of coins. Some people say they look like stacks of pancakes. On St. Patrick's Day, if you're eating green pancakes, maybe. Pennies can oxidize to be green. I don't know. These are called thylakoids. And they are also surrounded by a membrane. So there's actually 3 membranes in a chloroplast. So it's a 3-membrane system instead of just a 2-membrane system. In between the inner and the outer membrane, there's a space. What do you think we call it? Just like a mitochondria, we call it the intramembrane space. And then the space on the inside of the inner membrane, but not within the thylakoids, is not called the matrix, sorry. That would be too easy. It's called the stroma, but it's a fluid-filled space surrounding those thylakoids. Then within the thylakoids, there's also open space. I don't know if you can see that well. It's called the thylakoid space. And when you have a whole stack of thylakoids, it's called the granum. That's it. There's an R in there. If you have more than one of them, they're called the grana. There's a professor in the criminal justice department whose last name is Grana. When I think of him, when I think of chloroplast, or I think of, when I think of him, I think of chloroplast. So in this, where do you think the chlorophyll is located? Chlorophyll being the pigment that makes plants green. Where do you think in this structure of the chloroplast, where do you think it's located? Which part looks green? Yeah, so chlorophyll is in the thylakoid membranes. And that chlorophyll is what actually absorbs the light energy. Oh, shoot, I didn't mean to do that. Okay, so we talked about the basic overall equation. We talked about the structure of the chloroplast. Remembering this structure is going to determine function. We're going to talk about how that function occurs in the structure of the chloroplast. So again, we're going to start still with an overview. These are things, these diagrams, there's one for cellular respiration also. These are slides that I would print off and try to write on. Or pull up on your tablet and try to write on, try to fill them in as much as you can without looking at anything. Because these are really good overviews of what is going on. Now this should look kind of familiar to what we saw with cellular respiration. What do you see just without labeling anything on this? What do you see that looks like it might be something that we've seen before? We've got a circle. We've got ATP on there. We might have some kind of electron carrier, yeah. Okay, there's gonna be, again, a lot of similarities, but we're gonna see a lot of opposites. A lot of things that are happening backwards. So again, we're still in section 10.2. Okay, so we're gonna start on the left side. What do you see coming in here? Light, so this is what we call, this is what we call, don't write on it. There's two sets of reactions that happen. That have to happen to make photosynthesis, to make organic molecules. So the first set of reactions are the light reactions, because they need light. Okay, because they are using light, and we know that chlorophyll is what absorbs light energy, where do you think this is happening? Or you can tell from the picture. What did that look like from what we just talked about the structure? Happening in the thylakoids. So this is happening in the thylakoid membranes. And again, we're gonna separate, see how this reaction separates itself? This reaction, the light reactions are really taking water. Water's going through the light reactions, and what do you remember was produced by water from our overview? It's gonna produce oxygen. Okay, and this is sorry, there's something wrong. This is a separate process. This is where the photo part of photosynthesis comes from. And this, the point of this is really to split water. So as we split water and release oxygen, what's going to be left over? We're going to have hydrogen ions. And if we have hydrogen ions, what else should also be there? Electrons, because it's electrically neutral. Water is electrically neutral. So we're taking away the oxygen because that's one of the products, and we're going to produce hydrogen ions and electrons. When we have electrons bouncing around and hydrogen ions bouncing around, what does that remind you of in cellular respiration? Which part of cellular respiration has electrons moving around and hydrogen ions being pumped? Out of the 4 stages? There's a bunch of electrons and they're moving. And as we move the electrons, we're pumping hydrogen ions. The electron transport chain, which is part of oxidative phosphorylation. So what do we make in oxidative phosphorylation? What's the point of oxidative phosphorylation? To making ATP. So just like that, here we have electrons and hydrogen ions that are going to move through electron transport chains, really, to make ATP. Different than cellular respiration, we're not emptying electron carriers, we're actually going to produce some full electron carriers. So the electron carriers that we used in cellular respiration were what? There were 2 different ones that we filled and emptied. FAD, which we made into FADH2 when we filled it, and NAD plus, which we made into NADH. So very similar to that, we don't have any FADs in photosynthesis. We have NADPH is the electron carrier. So it's very similar, it just has a phosphate group. So that's a full electron carrier. So from the light reactions, we're going to split water. That water is going to produce hydrogen ions and electrons and oxygen. So this is all the stuff that came from water. These hydrogen ions and electrons have energy in them because they can move around and their movement produces energy. That energy is used to make ATP and to fill electron carriers. So at the end of this, do we have any carbohydrates? Have we made any carbohydrates at the end of the light reactions? Nope, not a single carbohydrate. Okay, remember when we had everything all lined up and we got to the right before oxidative phosphorylation, we made like 4 little ATPs. Okay, it's kind of like that. We've done a lot of work. We've gotten some ATP, we've gotten some full electron carriers, but no carbohydrates, which is the point of this reaction. So that's where the second part comes in. And it is a cycle just like the citric acid cycle. It's the Calvin cycle. It's named after one of the people that found it. 3 people figured out the mechanism for the Calvin cycle. Sorry, I want to write that up there also in words. So the Calvin cycle is the synthesis part of photosynthesis. We don't fully understand photosynthesis still. This understanding that we have of the Calvin cycle, they started to figure out in about the 1940s, which I know for you sounds like a long time ago because we're talking about the 1900s, but we're talking about less than 100 years ago that we figured this out. Okay, so if you were a biology student 100 years ago, you wouldn't be learning about this. Some of you are like, wish I'd been a biology student 100 years ago, and we haven't even gotten into it yet. Okay, this is taking place in the stroma. So this is the outer membrane. This is the inner membrane. So we're inside the inner membrane, but we're outside the thylakoids. So that fluid that's surrounding the thylakoids, inside the inner membrane is where this is happening. And this is where we're actually going to make carbohydrates. So we've used the water here. What's the other thing that we're going to use in this? The other reactant that we need to use here is what? Somebody said it. This is where the carbon dioxide is actually going to come into play. So we're going to go around the cycle. As we go around the cycle, we're going to put stuff together. So we're taking a low energy molecule and we're going to make a high energy molecule. So what are we going to need to put in? What do we have to do to make a low energy molecule into a high energy molecule? We have to add some energy. And where's that energy going to come from? What did we make over here? We're going to make it from, we're going to use up that ADP. So we're going to turn the ATP, sorry, into ATP, an inorganic phosphate. So we're going to break that bond. The bond in ATP is going to provide the energy that we need. And we're also going to empty our electron carriers, and the movement of electrons is going to help us get the energy that we need to synthesize carbohydrates from carbon dioxide. So we're going to have an empty electron carrier. So these reactions have nothing to do with light. So we call them in the Calvin cycle, the light independent reactions, or sometimes people call them the dark reactions. They can happen in the dark, but they're not generally happening in the dark because you need light for this part to happen. So it's just independent of light. It's not involving light. It's involving energy because it's using ATP, but it doesn't need light. It doesn't have that light energy. And here comes the physics. So much fun in one class period. Okay. So 10.3 starts us into the light reaction. So we're going to go into the details about the light reactions, which we're not going to get very far in today. And then we're going to talk about the Calvin cycle and the light independent reactions. So this is just showing us a little bit about how the chlorophyll functions. And so we have to go back to just basic concepts of light. So on the electromagnetic spectrum, this is, oh, sorry. If you can read my writing. Okay, so on the electromagnetic spectrum, we're talking about wavelengths of light that range all the way from like UV light all the way down to radio waves and that kind of thing. When we're talking about photosynthesis, we don't really care about anything except visible light, which has a range of about 380 nanometers to 700. And when we talk about visible light, what is the acronym that we use for the different colors? Roy G. Biv, red, orange, yellow, green, blue, indigo, violets. There's your Days of the Magic School Bus. We have a Magic School Bus, Roy G. Biv book. So sunlight is producing not only visible light, but we're only caring about the visible light because that's the only light that's useful for photosynthesis. So you can see as light comes from the sun, it's white, but we know that that white contains all of the colors, the Roy G. Biv colors. Some of that light is absorbed by the chlorophyll. And some of that light is either transmitted, like it keeps going through, or it's reflected. So when we talk about light coming in, visible light and chlorophyll, chlorophyll is going to reflect green light. And it's going to absorb best the reds and the violet blues. That says blue, wow. Just like my sweater is yellow, all the colors are hitting this. It's going to absorb everything except yellow, and it's reflecting yellow back at you. The green leaf is absorbing everything except green, and it's going to reflect green back at you. Okay? Except when fall happens and chlorophyll starts to break down, and we start to see some of the other pigments, like the reds and the oranges and the yellows or the browns, if you're an oak leaf and you don't really want to look pretty. So this is kind of what it looks like when we talk about absorption and the absorption of different pigments. There's different kinds of pigments besides chlorophyll. Chlorophyll A, there's actually multiple kinds of chlorophyll too. Chlorophyll A is the most dominant pigment. It's what we call the primary pigment. And you can see it's kind of a greenish blue color. Chlorophyll B and the carotenoids are what we call accessory pigments. So they're not responsible for the majority of absorption of energy, but they can absorb some energy, and you'll notice they absorb at different wavelengths. So we said that chlorophyll, that green, or that photosynthesis happens best when red and violet blue are absorbed, and you can see here, here's our spectrum of visible light. So it's actually going the opposite way, Vib, G, Yor, I don't know how to say that backwards. And you can see there's a peak here in the violet blue, and there's a peak here in the reds where the maximum absorption happens. Chlorophyll B is going to absorb a little bit differently, more in the blues and a little bit in the oranges. And then the carotenoids are going to absorb also in the blues the most. They're the orange color. So a lot of maple trees have a lot of carotenoids, and that's what we're starting to see now is the orange color. There's a really pretty one on the way to Chapel that has a nice orange color. Carrots are pigmented with carotenoids, which actually have a function in your eye, which is why eating carrots can be helpful for your eyes. It's not just a myth. The primary pigment is useful mostly for photosynthesis, absorbing energy for photosynthesis. The accessory pigments are mostly used for photoprotection. So they're protecting chlorophyll. Chlorophyll can be damaged by too much light. So that's why one of the reasons that plants have different pigments is to protect the chlorophyll from getting damaged from too much light. So light comes in, it's absorbed or reflected. The stuff that is absorbed allows photosynthesis to happen. This one up here is an absorption spectra, so it's showing you where absorption happens. This is the action spectrum, so this is measuring the rate of photosynthesis. So at what wavelengths does photosynthesis happen the best? And you'll see it corresponds pretty well with where chlorophyll A is absorbing its max absorbances. So photosynthesis happens best here in this purple range. and this red range. So what color light should you shine on a plant if you want it to grow best? Purple lights or red lights? What color should you not shine on a plant? Green, because that's not where photosynthesis is gonna happen. That's not where the most absorption happens. So if you look at a plant light, if you go on Amazon to buy a plant for your favorite a light for your favorite little plant, it's gonna be a purplish blue color, or it's gonna be the ones that have red and blue both. The LEDs that have both red and blue, because that's the most effective light color for your plants. So the most effective wavelengths are what's gonna make photosynthesis happen. So all of this is happening when this light comes in and is absorbed. Oh, wow. I didn't know I could do that. Okay, I can still do it. It won't let me write them. So what that light is going to do is it's going to actually excite the chlorophyll molecules. So it's going to excite electrons in chlorophyll. So the light that gets absorbed will excite the electrons in the chlorophyll, and it's actually going to cause them to move. Okay, so light's coming in as a photon. I don't know if you remember from physics, the whole wave particle duality theory of light. That's what I remember from physics, the wave particle dualities. Light is a wave, but it also functions as a particle. Those particles are called photons. So as the photon comes in, electrons are at their ground state. That's their normal orbital. They can absorb the light energy and they can move up to the excited state. So that excited state in chlorophyll is going to be moving it up to an orbital with a higher potential energy. So it's going to excite it and move it up to an orbital with higher potential energy, meaning it's further from the nucleus. This will make it not stable. So this is temporary. So it's in this excited state and it's going to fall back down to the ground state. And when it does, it's going to release heat. And it might also fluoresce, releasing a light particle itself. This is happening in isolation. So this is if we take chlorophyll out and put it in solution. We will talk about what happens when it's in the process of when it's in the actual normal state of a thylakoid membrane. Okay, staff will remember you can get extra credits. Look at the assignment on Moodle. Quiz on Friday. And we will get to the light reactions on Friday for real, I think.